Today, we are going to talk about a story that would have sounded like pure science fiction just a few years ago. Imagine a very real situation. A person suffers a heart attack. Doctors manage to intervene in time. Everything goes well. Their life is saved. It seems like the worst is over, right? But in reality, deep inside the chest, irreversible processes have most likely already begun. Why does this happen? And more importantly, how is modern science trying to literally crack and reprogram this seemingly unavoidable fate? Let's find out. So, let us go back to the very first minutes after a myocardial infarction. This is not simply a temporary system failure. Within minutes, a large portion of the heart can be deprived of oxygen, and heart muscle cells begin dying at an alarming rate. Naturally, the body reacts immediately. It throws every available resource into damage control in an attempt to save the situation. But here is the paradox. That emergency response creates a dangerous trap. Instead of carefully rebuilding the damaged tissue, the body switches into an emergency patch repair mode, and that process almost always ends with the formation of a permanent scar. At this point, a perfectly reasonable question arises. Why does a cut on the skin often heal so well that we barely remember it was there? Why can the liver regenerate from a remarkably small remaining portion? Yet the heart, our most important tirelessly working engine, seems almost incapable of repairing itself properly. It is one of the most fascinating mysteries in biology. To appreciate the scale of the problem, let us compare healthy heart tissue with scar tissue. Healthy cardiac muscle is a marvel of biological engineering. It contracts with tremendous force and conducts electrical signals with extraordinary precision. Scar tissue, on the other hand, is essentially a patch. It cannot contract. It cannot pump blood. And perhaps most importantly, it behaves like an electrical insulator, blocking the signals that normally travel through the heart. Slowly but surely, this inactive region forces the remaining healthy muscle to work harder and harder. So why is the heart so resistant to regeneration? The answer lies within the heart cells themselves, the cardiomyocytes. These are not ordinary cells. They are highly specialized biological machines. Inside them are densely packed protein structures called sarcomeres. Their entire architecture is optimized for one purpose and one purpose only, contracting continuously and rhythmically throughout our lives. And the problem is largely mechanical. For a cardiomyocyte to divide, it would first need to dismantle much of this incredibly complex internal machinery. But these cells were designed for performance, not reproduction. And this is where researchers encountered a surprising discovery. The most intriguing part of the story is that surviving heart cells actually do attempt to divide after injury. They begin the process. They successfully replicate their DNA. They increase their chromosome content. And then suddenly, they hit what appears to be an invisible concrete wall. The process breaks down at the very last stage. Biologists call this barrier cytokinesis. This is the final step in cell division, when one cell physically separates into two independent daughter cells. And this is precisely where cardiomyocytes fail. Unable to complete that final separation, they become enlarged, multinucleated cells. No new functional heart cells are produced. And without new cells, there can be no meaningful regeneration. So what can be done? Science has found an astonishingly elegant workaround. Let us rewind to 2006, when Japanese scientist Shinya Yamanaka made a discovery that transformed biology and eventually earned him the Nobel Prize. He demonstrated that mature adult cells could be reprogrammed back into an embryonic-like state. Remarkably, this required only 4 proteins, now known as the Yamanaka factors. OCT4 helps cells forget their specialized identity. SOX2 promotes cellular renewal. KLF4 supports cell division. But the cocktail also contained one particularly dangerous ingredient, c-MYC. c-MYC is an extremely powerful oncogene. It dramatically accelerates cell proliferation, but it also carries a significant risk of cancer development. And this brings us to the major breakthrough described in a new study published in June 2026. Researchers found a brilliant solution. They simply removed the dangerous component. No c-MYC. Only 3 factors remained: OCT4, SOX2, and KLF4. The resulting combination became known as OSK. It retains the regenerative potential of cellular reprogramming while eliminating the most concerning cancer-related risk. This development opened the door to genuine clinical applications. The elegance of the new approach lies in its subtlety. Researchers did not attempt to transform heart cells back into fully pluripotent stem cells. That would essentially erase their cellular identity. Instead, they used a strategy known as partial reprogramming. OSK therapy gently relaxes the rigid sarcomere architecture for a limited period of time. The cell gains enough physical flexibility to complete division. Yet genetically, it still remembers exactly what it is. It remains a heart cell. In many ways, this represents a complete shift in scientific thinking. Previously, researchers believed the goal was to force dormant cells to divide. OSK suggests something entirely different. Perhaps we do not need to force cells at all. OSK simply removes the obstacle at the cytokinesis stage. The cells that were already trying to divide after a heart attack can finally finish the process they had already begun. We are not pushing them. We are simply clearing the path. And in practice, the results have been extraordinary. Experiments in mice exceeded expectations. When animals that had suffered heart attacks received OSK therapy, their hearts became significantly more effective at pumping blood. Researchers documented measurable reductions in scar size, but perhaps the most remarkable finding was that entirely new, healthy cardiomyocytes appeared precisely within the damaged region itself, not elsewhere, exactly where they were needed. When compared with previous regenerative strategies, the difference is dramatic. For years, scientists placed enormous hopes on stem cell therapies. Unfortunately, transplanted stem cells often survive poorly within damaged heart tissue. Researchers also attempted to directly reprogram scar tissue cells. The results were largely disappointing. OSK takes a fundamentally different approach. It works with the heart's own cells, unlocking a regenerative ability that was already present. Yes, the timing must be controlled with extraordinary precision, but the therapeutic potential exists on an entirely different level. Interestingly, these remarkable laboratory findings align closely with ideas proposed by Harvard biologist David Sinclair. Sinclair is an Australian biologist and professor of genetics at Harvard Medical School, widely known for his work on aging and longevity. He proposed what is known as the information theory of aging. According to this theory, cells do not simply wear out. Instead, they gradually lose access to the information that allows them to function as youthful cells. In this framework, aging is not merely hardware deterioration. It is more like software corruption. The information still exists. The cell simply loses access to it. It is as if every cell contains a backup copy of its ideal state. And if access to that backup can be restored, the cell may once again function properly. Why is this relevant to the heart? Because partial reprogramming appears to reactivate that biological backup system. It gives cells access to information that allows them to repair and renew themselves. Perhaps the most encouraging aspect of all this is that it does not appear to be a one-time success limited to a single organ. Instead, it represents an entirely new direction in medicine. The concept of restoring a cell's biological backup system has already shown promise in other tissues. Similar approaches have been explored in vision restoration, in the brain, in the skin, and now, in the heart. Looking ahead, it is not difficult to imagine how transformative this could become. Today, the best medicine can do after a heart attack is restore blood flow and slow the progression of heart failure. But tomorrow may look very different. Tomorrow, physicians may be able to deliver a temporary OSK therapy directly to the damaged region and stimulate the growth of entirely new heart muscle. That is no longer symptom management. That is true tissue restoration. Of course, researchers remain appropriately cautious, and for good reason. The greatest challenge is timing. If reprogramming remains active even slightly longer than intended, heart cells can become excessively immature. They may lose their ability to contract effectively. In the worst-case scenario, this could compromise heart function or even trigger uncontrolled cellular growth. That is why precise control of this therapeutic window remains the highest priority for researchers today. And while scientists continue working to make this approach completely safe, one profound question remains. What if the heart never truly lost its ability to heal? What if it simply forgot how? And if we now know how to remind cells of their youthful potential, could the day come when a heart attack is no longer viewed as a permanent sentence? It certainly feels as though we may be standing at the threshold of an entirely new era in medicine. And perhaps in the future, a heart attack will no longer be considered a point of no return. Take care of your health, and I will see you in the next video.